All right. Well, hello everyone, and thank you for joining this special journal club for our special issue of March 2023. I am Pedro Ramirez, editor-in-chief of the International Journal of Gynecological Cancer. This has been really an amazing and really very impacting special issue. I will share with you some of the metrics that we have already been getting with regards to the downloads of these articles. So I want to thank you all for joining and joining in on this discussion. As traditionally has been for the special issue, we have the guest editors that are invited to discuss the articles that were included in this special issue. And I should say that for this special issue, we aim to have very high quality articles as evidenced by all the downloads that you have seen, and we will share with you. And by doing so, we asked specific individuals, leaders in gynecologic oncology, leaders in the topics that were published in this special issue to give us their update as it pertained to these very important topics. So before we get into the articles, I want to introduce Ainhoa Madariaga and Dr. Rob Coleman also, who are joining us. Ainhoa, I'll start by asking you to just say a little bit about yourself, and then we'll follow up with Rob, and then we'll get into the discussion.
Hi, thank you so much for the invitation. For me, it was an absolute pleasure to be an editor of the special issue, to be involved in the selection of the topics and the authors. So it was great to be part of it, and it's amazing to see the outcome and how good it turned out to be. So I'm Ainhoa Madariaga. I'm a medical oncologist specialized in gynae cancers. My areas of interest are drug development and gynae cancers. I work at 12 de Octubre University Hospital in Madrid, Spain. And I also serve as the chair of the Young and Early Career Investigator Group of the gynae group of the EORTC.
Well, thank you so much, Ainhoa. And I think that when we met many years ago in Canada, I really saw. I said I'm really meeting a superstar in the field of gynecologic oncology, and you have continued to really shine. And I'm really grateful for you taking part of this. I know this is a lot of work, so thank you for being a guest editor on the journal. Really very exciting to see all the work that you do and all that you are contributing to gynecologic oncology. So then our second guest editor, who is in the journal club, Rob Coleman, needs no introduction, but certainly Rob, would if you could just tell us a little bit about you and yourself. And I can also share that the minute I met you many more years back, I definitely knew I was meeting a superstar in gynecologic
That's
oncology.
painful. As I say, my name is Pedro Ramirez, and I'm so glad to be with you. Oh, wait a minute. Oh, different script. No, thank you guys. It's so awesome to be with you. And just seeing all these based on the screen is just, oh man, this is why we do what we do. I just love it. It's been an honor to work with Ainhoa and Antonio to put this together. It's been a wonderful project. Obviously, the quality of these papers has been amazing. No wonder there's a lot of downloads. But so I'm Rob Coleman, G1 oncologist here in Houston, with where Pedro is also resident, and I serve as the chief medical officer for the SCRI, which is a site management organization for clinical research across the country. Very happy to be able to work in my absolute clinical passion, which is in clinical trial, drug development, and education. And so being in front of you all and working with you and has just been an absolute pleasure, and it's actually what makes me get up in the morning. So I'm really excited to be here, and thanks for giving me the opportunity to work with you.
Excellent. So much. Thank you, Rob. And also, I should say thank you to Antonio Gonzalez, who couldn't be with us, but who also was a guest editor. So I know that, certainly we have now over 60 participants joining in on the Journal Club. We want to give them opportunities to ask questions. But I wanted to just go over some of the really important manuscripts that were published in the special issue. And I should add, all of these are actually free for downloading as well. I want to start with the first manuscript that I think is a really important topic to discuss, particularly as we look at evaluation of outcomes in clinical trials. And this is titled Outcomes and Endpoints of Relevance in Gynecologic Cancer Clinical Trials. It has been downloaded so far this month, and certainly in the short span of 3 weeks, 983 times already.
Oh my gosh, really?
So this is really remarkable.
And they weren't all me. That's what's amazing. It wasn't just me.
That's right. That's how many times Rob compressed that button. But I wanted to start with you. I know you're the first author on this article. Why is this relevant? Why do we need to be more critical about evaluating outcomes and endpoints for, particularly in given that there's so much research being done now looking at oncologic outcomes?
So as part of the paper, we wanted to highlight the heterogeneity on choosing the primary outcome of a study, and not the same outcome is the best for all type of studies, all type of drugs. And as part of the paper, we included you to have the surgical viewpoint. We included Fernanda from Lausanne to have the radiation oncology viewpoint. And it was great to integrate medical oncology, radiation oncology, and surgical oncology and see what makes sense in each scenario. As part of the paper, we highlight that it's true that overall survival and quality of life are the gold standards. That's what we want for our patients. We want them to live longer and live better. That's clear. But sometimes, depending on the scenario, another endpoint may be better to be chosen as a primary endpoint, especially depending on the type of disease and the type of agent. And that's what we discussed there. We discussed PFS as primary endpoint, the choice of intermediate surrogate endpoints like PFS2 and time to second subsequent therapy, which has an important impact in maintenance therapies such as PARP inhibitors. And we also discussed the importance of overall survival as a primary endpoint in certain clinical trials or certain drugs such as immunotherapy.
Great. And I know you mentioned this PFS2, and I see that coming up more and more frequently. Can you talk a little bit about that? Because, obviously we're always used to, following the progression-free survival, but now what is this progression-free survival 2? So
with some of the agents, like for example, PARP inhibitors, we know that they prolong the progression-free survival. They increase the time, like since you start the maintenance treatment to the moment where the disease progresses. But we want to know what happens after, because certain treatments may give some resistant mechanisms that make that some other drugs work less well, or you may have some side effects, like for example, in recurrent setting, PARP inhibitors have been associated with myelodysplastic syndrome and hematological malignancies. So we want to see, although percentage is very low, so we want to see what is the impact after the treatment, after you progress on the treatment. And this kind of secondary endpoints, such as PFS2, give us the time on the subsequent treatment. And with PARP inhibitors, that's what we have seen in the maintenance setting. PFS1 is increased, but PFS2 with the subsequent treatment is also increased with the treatment. So we see that the benefit of the treatment is prolonged over time.
Excellent. Well, thank you so much. I know, Rob, anything else you want to add with regards to the importance of this evaluation?
Oh, I think it's critical. Think about where we are in our current standards of care today. The way we continue to change the standard of care is to conduct these trials. So the clinical trials is absolutely critical to the to continuing to move the needle, as we always say. And I think that as along the way, these endpoints are becoming scrutinized now because they're becoming more molecularly annotated. And so it just makes our need, and our abilities to develop, really informative clinical trials so that we can reduce sample size, not expose women to potentially likely to benefit them type of therapy. And so it's again, as we get better as being clinical trialists, these questions can be addressed in a more rapid fashion and with clearer results.
Yeah. And Rob, I just wanted to follow. I mean, for many years, we have been focused on, you got to prove that there's a benefit in overall survival. And, that's like that's the milestone you want to reach in any clinical trial. given the fact that at recurrence, particularly in ovarian cancer, there's like so much branching of what you can get, shouldn't we just focus on progression-free survival and not so much on overall survival?
So you're baiting me. I get it. I get it. Yes, of course, I'm super passionate about this. We have an editorial.
For many of our international listeners, baiting means when you throw the hook to a fish and you start going like this.
So we feel very strongly that that if you're doing a lab experiment, you would take your subject, you would do an intervention in one of the dishes and none in the other and see what happens, and you would be looking for the immediate outcome. And that's really important for us to understand what's going on. One of the reasons PFS2 is, as you mentioned before, is becoming more you're starting to see it more, is that it's not a bad determinative of like the persistence of the benefit seen in the previous line of therapy. So just as you mentioned, after the second additional line of therapy, it starts to branch out quite a bit, and we don't control for any of those variables. So there's a couple ways you can address this. One is you can define, let's say, therapy number 1 and therapy number 2 and therapy number 3, and then account for, how that would play out. Because as you add another node, you basically square the number of potential opportunities there. So that's that one way to do it. The other is to design gigantic trials that will be able to assess, all of these confounding variables. And neither of them are really all that helpful, because as you overpower for overall survival, as you just mentioned, you can detect very small differences in progression-free survival that are not really clinically meaningful. Mm-hmm. So we have to strike this balance. And I think the reliance on overall survival, which many people say, hey, listen, it's an unbiased endpoint. Well, that kind of is true, but it's also kind of not true, because influenced by multiple different factors along the way that could change, including things like surgery. Or if it's been so long, a new intervention could have become available, like immunotherapy. And so we would have been able to account for that. And that's one of the reasons why we're so passionate about the earlier time points.
Fantastic. So Rob, I'll stay with you. And this next topic, really very, very relevant and very hot right now. Updates in the molecular classification of endometrial carcinoma. This is an article published by Alicia Leon Castillo. And I should say that in a span of 2 weeks, in 15 days, it's been downloaded 1,834 times. So
amazing.
Well, this is a great time to be talking about endometrial cancer. For years, during from our fellowships, yours isn't that far away from mine. Not that many fellowships ago, we didn't have anything to talk about in endometrial cancer. It was chemotherapy and hormones, right? So now that we've learned that since 2013, that there are these molecular characterizations that identify our POL-E mutations, our MSI patients, patients that have copy number high and copy number low, that kind of characterization now being further subdivided into specific molecular characterizations is now allowing us to be much more precise in what kinds of therapies we can use. But even more importantly, is we've gained the interest of the pharmaceutical industry to help us bring some of these therapies that have efficacy in other tumor types, but have similar genomic signatures in endometrial cancer. And as you just mentioned, as it relates to immunotherapy, we had a couple early successful wins with immunotherapy, showing that patients that have microsatellite unstable or deficient MMR reflected cancers were responding to single agent immunotherapy. That was very exciting. We took what we knew about anti-angiogenesis and combined it with immunotherapy as another particular strategy, highlighting the patient population that did not have that molecular signature of MSI. And we got that drug as an accelerated approval. And then we moved them into earlier lines of therapy. And so as you may know, next week or so, we're going to have 2 presentations at SGO, accompanied by 2 New England Journal of Medicine articles. So how positive those trials are, that are basically showing that when we add immunotherapy to chemotherapy in patients with advanced stage or first recurrent endometrial cancer, we're actually improving their outcomes. And it's so exciting to be able to have that therapy now moved from a single agent for recurrent patients all the way into the frontline setting. And we have more trials coming that are even looking in the adjuvant setting for high intermediate risk, non-metastatic patients. So we would have never done any of this if we hadn't done the clinical trials that we talked about before and understood the molecular characterization so that we could actually take advantage of that.
Fantastic. So looking forward to those SGO presentations and subsequent publications. Now, I know let's go to the topic of, there's an article on immunotherapy in endometrial cancer. What is the most promising immunotherapy as you see it today for endometrial cancer?
I think that anti-PD-1 treatment is the most promising right now. At the recurrent setting, as Rob mentioned, we have a pembrolizumab and dostarlimab for patients that are MMR deficient, and then for the overall population in Europe or MMR proficient in the US, we have the combination of pembrolizumab and lenvatinib. So those are really great drugs that work well, that give durable responses to our patients. And then moving to the front line, as Rob mentioned, we'll have 2 studies assessing the role of pembrolizumab and dostarlimab along with chemotherapy with a maintenance treatment subsequently. And I haven't seen the results, so looking forward to reading those 2 New England Journal articles next week and seeing what they show. But
And just as a special announcement, I heard Rob is just going to tell us the results now before the presentation and before the New England publication. So Rob, please go ahead.
Did you get that?
Totally. You know there's going to be people slowing down the video to see to do
your mouth.
Lip reading, yeah.
Exactly. So I know I'll stay with you because this article, it was published by your former colleagues, Stephanie LaRue and Pam Sobrenares Pina, overcoming PARP inhibitor resistance. What is PARP inhibitor resistance, and how do you overcome that? So we know that most patients that are on PARP inhibitor for a long time, like nearly all of them in the recurrent setting, most of them in the frontline setting, will develop some kind of resistance to the treatment. And one of the challenges is that resistance to PARP inhibitors is usually also associated to resistance to other kind of drugs, such as platinum. The most common mechanism of resistance, or maybe the better known mechanism of resistance, is reversion mutations in BRCA, which makes the protein is working well again, so the drug doesn't work as well. And there are many other resistance mechanisms that we know well, such as amplification in cyclin E and other reversion in other HR-related genes, amplification of the drug efflux of the gene that encodes the drug efflux pump that brings out the olaparib and the Taxol from the drug. So there are several resistant mechanisms that have been described in vivo, like in our patients. And now we're trying to learn how to overcome that. And to do so, there are several studies that have been developed assessing novel combinations. In some of them, they are associated PARP inhibitors with anti-angiogenics, ATR inhibitors, like targeting the cell cycle, ATR inhibitors, GLUT1 inhibitors, and other kind of drugs, and also trying to improve the way the drug is delivered with antibody drug conjugates. So there is a lot of studies in PARP inhibitor resistance, because that's our present. Nearly all our patients with high-grade serous ovarian cancer are receiving PARP inhibitors. So we need to go ahead and know what to do after the resistance mechanism arises. Great. And Rob, any comments with regards to how you manage your patients that have that PARP inhibitor resistance?
Yeah, I think, as was mentioned, I think that, we're trying to understand exactly what that means. So as our patients are moving, are getting their PARP inhibitor exposure in the frontline setting, not all of them are progressing while they've taken the PARP inhibitor. So we have patients that have PARP exposure and then have a relapse, and those that have PARP progressors. And I think that there's, we're learning more about what those mechanisms are. But as we learn more about what might be leveraged at the cancer cell level that's escaping this kind of strategy, we've gotten to be much smarter about how to do these combinations. And so, just as Ana mentioned, the idea of doing these novel combinations and designing the trials that make them informative. Again, we get back to that very first thing you mentioned, how do we design great trials to answer these questions? We're starting to come up with really neat and novel strategies to assess these new combinations that could overcome that resistance mechanism.
Fantastic. So now, Rob, we also have a couple of articles on rare ovarian tumors.
Yeah.
So I'll start with low-grade serous carcinoma. And, for many, many years, the only option was, carbotaxol. And we're not that enthusiastic. In fact, we're still not that enthusiastic about that recommendation for those patients after they undergo surgery. And, the next option is hormonal therapy. But now it's like some really very exciting options for patients with low-grade ovarian cancer. Can you just tell us a little bit about that?
Yeah, thanks so much. This is another disease type that has benefited from the combination of really expert pathologists, smart clinicians, and molecular annotation. And so what we've learned, just as you mentioned, that we tried a bunch of traditional therapies that didn't seem to work. Our pathologists and our molecular geneticists have told us that this is a different cancer. Unfortunately, it shares the name, grade and serous. But they are different diseases, and it's taken us quite a bit of time. This year, I've seen the first kind of acknowledgement that this is actually a rare tumor. It used to be just thrown in with the other ovarian cancers, but the natural history and the biology are just so different that this gives us an opportunity to investigate new drugs that really don't work in high-grade serous ovarian cancer. And some of those are we're listed in the article, such as MEK inhibitors and BRAF inhibitors, the combination of the two. And then, utilizing strategies to move those types of therapies in the place of chemotherapy. And we're able to do actually an ongoing frontline trial right now, which is comparing hormones followed by as adjuvant treatment followed by hormone maintenance versus chemotherapy. So followed by a hormone maintenance. So we have this opportunity to really change the landscape of how we approach this disease. Again, highlighting that molecular annotation is absolutely critical to make sure that we understand, what's going on in the tumor. And I'll just leave it with one other kind of really cool observation. So these molecular targets are starting to become even subdivided. So one that we see frequently is a RAS mutation. In lung cancer, RAS mutations that carry a specific G12C mutation, there are drugs directed specifically for that mutation. And that has shown merit with 2 different drugs that are available for lung cancer patients right now. And coming soon, there are G12D, a more common RAS mutation phenotype. So in addition to just having a class, we're now getting down to the really nitty gritty of how we might be able to leverage what we're learning from other tumor types for this specific tumor that has a frequent alteration of that pathway.
Fantastic. And I want to encourage everyone to submit their questions, and there are questions that are coming in, and we definitely are going to get to some of those questions. I wanted to just hear from Ainhoa with regards to another rare tumor, clear cell carcinoma. Clear cell is one where I think traditionally it's just been, is carbotaxol like everybody else. So anything that looks exciting for clear cell carcinoma?
I think that immunotherapy may have a role in clear cell carcinoma. Anti-PD-1, anti-PD-L1 inhibitors, they have shown some signal of action. And if we look at the studies at the recurrent setting, probably the clear cell carcinomas were the ones that had better kind of response to immunotherapy, to single agent immunotherapy in this setting. So there are several studies ongoing looking at those agents. On the other side, we also know that knowing better the biology of clear cell carcinoma, we know that nearly half of them have ARID1A alterations. And recently there have been some studies also showing signal of activity of ATR inhibitors in clear cell carcinoma and in patients that harbor ARID1A mutations. So we're learning more from the biology of these tumors and then trying to look for different treatment strategies.
Great. So actually, before we leave the topic of ovarian
cancer, and anti-angiogenics. Anti-angiogenics work great also, the bevacizumab.
Exactly. Let's not forget that. But certainly I want to take advantage of having both of you on the panel. And also I want to thank actually patients that have joined this discussion as well. So one of the questions actually I think is a very important question. And Brenda asked, I have high-grade serous carcinoma, just started on a PARP inhibitor. It's very concerning that I go through some of the side effects and I may not see benefit, and may also develop resistance to other treatments. It seems that, certainly from the title, I can understand that success depends on which genetic mutation I have. And we don't often have the ability to know. So taking the medication, potentially going through these side effects, are we flying blind? That is her question. Rob, you want to start with that?
Yeah, it's a good question. I think, we're learning more about, in whom these therapies work. So it really kind of depends on which line and setting and previous exposure. In the what we have seen is that the alliance between a biomarker such as a BRCA mutation in the tumor, or in a tumor that doesn't carry a BRCA mutation but carries a signal that reflects homologous recombination inefficiency or deficiency, that the PARP inhibitors do show a benefit, and the benefit is not little. So in a way, without any information, about half of our patients are potential good candidates for this therapy. Now, the question is important also because if we do know what's going on in the tumor, the question is what alternatives do we have to a PARP inhibitor in the what we call the HR proficient or the test negative tumors, and that's actually an area where we're actively studying. We certainly do have access to drugs like bevacizumab and PARP inhibitors in that setting in certain situations around the world. But it does, for me, bring up that, hey, listen, this is a situation where the drug is very specific for an alteration, and if the alteration is not there, we don't really have a lot of expectation it should work. So we should use it in the negative as well as the positive. So again, the idea of not taking therapy because of the potential development of mutations or development of resistance is hard to it's hard to square, because we don't know what's going to happen in the future. We don't know that everybody's going to develop a resistance mechanism. And what we try to do is to take the information we have at hand, the best information we have at hand, and provide the best opportunity for our patients at that time. And right now we assimilate that data and try to provide as much information to use as much information to inform our decisions as we can.
Yeah, great. Thank you so much, Rob. And another question, actually, I know I'll address this one with you. Wondering whether there are any predictors after finishing surgery and chemotherapy that would indicate that the patient's likelihood of recurrence is so low that they may not need a PARP inhibitor at all, or any adjuvant treatment at all.
So it's like, for me, for most patients, like nearly all patients with a stage 3-4 disease and a high-grade serous carcinoma, I would give them maintenance treatment. Like there are very, very, very few scenarios where I wouldn't give maintenance treatment in this setting. Most cases, if it's HRD, BRCA mutation carrier, they will receive a PARP inhibitor for sure. If they are not HRD, well, there are different options in this case. Sometimes I would give bevacizumab. Sometimes I consider a PARP inhibitor also in these cases. But for a high-grade serous carcinoma, even though they do great with chemotherapy and great with surgery, for me, it's very, very stressing not to give adjuvant treatment. I always use it. Maintenance treatment, sorry. Yeah.
Very well. So we'll get into another question now, this one addressing endometrial cancer. How do you both treat post-immunotherapy progression endometrial cancer patients?
Easy. That's so easy. Let's move on. No.
I'll start with you, I know, and then we'll let Rob get ready to answer that.
So this is a tricky setting. There are some studies, like there is like very, very few studies seeing what to do after immunotherapy. There is a small study from the Princess Margaret Consortium where they gave nivo and a cabozantinib combination, TKI plus immunotherapy in the post-immunotherapy setting, showing an interesting signal of activity. I think that there may be a role of retreatment with immunotherapy with different combinations, but we need clinical trials for that. So the best treatment option for sure is a clinical trial. As part of the standard of care, depending on the molecular subtype, I may give different treatments. So imagine low-grade endometrioid carcinoma, slowly growing, hormonal receptor positive, probably I would treat that patient with hormonal therapy. If it's a serous p53 abnormal carcinoma, probably I would try to get clinical trials with WEE1 inhibitors or some kind of treatment like that. So we need to look at the specific molecular subtype, and probably clinical trials is the best treatment option. We know that response rates are 10–15%. So for sure, a clinical trial is the best.
Fantastic. And Rob, I want to take advantage of your time, and Rob has now exactly 10 minutes from going on to his next meeting, and he tells me he can only run 3 meetings at the same time, so we don't want to interfere with that. But Rob, I want to ask you about an
One on each.
Yeah.
One on each device: iPad, iPhone, and computer. I only can run 3.
This is a really important article, very good article, and I highly recommend it as well. Written by you and your group, Anka Kelaruz, Van Manner, Katie Moore, and Keta LaRusso, and yourself, on antibody drug conjugates in the management of gynecologic cancers. Tell us as to why these are important and what's looking promising.
Yeah, this is a great new therapy that's kind of entered our space, and not just UIN, but across many solid tumors. The antibody drug conjugates, not a new topic. It's just that they've gotten so much better, so much more stable. And these are essentially antibodies that are targeted to specific antigens or surface molecules on ovarian on cancer cells, and then uses that connection between the receptor and the antibody to bring this compound into the cell, the cancer cell, break out the warhead or the chemo component of that, which can then cause cell death in a very selective way. So these are exciting because they're kind of like smart bombs. We can use a lot of what we know about the array of how cancer cells are different than normal cells, and we can take advantage of that to design these really cool molecules. Currently, they're basically mostly focused on a single antibody and a number of chemotherapy molecules that are the same. But this field is moving. So we now have these bispecific type of antibody drug conjugates that have one arm is focused on one kind of target, and one arm is focused on a different kind of target. So now we are able to even take advantage of heterogeneous expression of what we see in the cancers to deliver these highly toxic chemotherapies. And now we're also adding different types of drugs. So it's not just chemotherapy warheads, but other types of things that can have impact on the cell. So this platform, I like to think of them as platforms, not just as about a drug. This is a platform that is just at the tip of really kind of expanding into completely new markets. And so this offers a lot of hope, I think, for our patients.
And Rob, how far are we from this being accessible to countries other than obviously United States and Europe?
Yeah, so that's a good question. Many of these drugs are working their way through the various different health authorities. Our 2 most mature drugs are one focused on cervical cancer, so sotrovimab vedotin, and one on ovarian cancer, which is mirvetuximab soravtansine. But there's a whole bunch of drugs that are coming behind them to help kind of get into these new disease states that can then, again, with support, get them into multiple markets around the world. So we're starting to see them kind of make their way, including, these our pharmacological sponsors are leveraging regulatory strategies where they can get multiple country approvals under the same trial.
Fantastic. Well, Rob, thank you so much. We want to give you about 5 minutes in between your meetings, so truly appreciate you being part of the discussion. And we'll finish up the journal club with Ainhoa Madariaga. But really, truly
I hope I
fantastic
can
job.
survive without Rob.
No, that's all good. Oh my gosh. Well, I'm so pleased to be with you, Pedro. Thank you so much for inviting us and me, and thank you for letting us do this special issue. It's a great it was a great piece of work, and we have so many great authors, and I'm hopeful it'll help.
Oh, fantastic. Well, have a good rest of your day, and thank you
You
once
too.
again.
All right, guys. Thank you. Bye-bye.
I know I wanted to now talk a little bit about cervical cancer. And if you can talk to us a little bit about what's going on with regards to immunotherapy and targeted therapy for cervical cancer.
So we're at a great time for cervical cancer with recent approvals of immunotherapy in this setting. As you know, in the frontline setting, until very recently, we had chemotherapy, platinum-based chemotherapy that we have been using for a long time. Some patients could benefit from anti-angiogenics, but then we have the issue of possible fistulas and side effects like that are not easy with the bevacizumab in certain cases. And now the immunotherapy came here to stay. We have pembrolizumab in the frontline setting. It showed that it increases both PFS and overall survival, which were the co-primary endpoints of this study. At 2 years, the 10% of patients were alive. Like the difference between groups was 10% higher, favoring the pembrolizumab arm for overall survival. So that's a big achievement for cervical cancer. It's a disease that didn't have many treatment options until now. And now with immunotherapy, we're changing the treatment landscape, and we're able to provide our patients with treatment options that work well and have low toxicity. And we're seeing really these durable responses. We're seeing patients that are on immune treatment for years. So that's really great.
And I know one follow-up question, and often, patients will ask with regards to, obviously, we are now routinely looking at a combination treatment, but the PD-L1 status in the recurrent cervical cancer patients, certainly many patients will ask, and well, even if I'm PD-L1 negative, why can't I have immunotherapy as well? What do you say, and what's your discussion with those patients?
Yeah, so on frontline setting, unfortunately, the approval for pembrolizumab both in Europe and in the US depends on the PD-L1 status. Only patients that have a CPS equal or more than 1% are able to get the pembrolizumab. But on the recurrent setting, the approval for cemiplimab is not like that. Patients that have PD-L1 negative can also have access to cemiplimab in the recurrent setting. So there is an option to treat those patients in the recurrent setting with immune treatment. And we also try to look at other potential biomarkers. Like, for example, if you do an NGS panel and you see that the patient has a TMB high, a high mutational burden, well, those patients are also going to benefit from immunotherapy. And we see some patients with cervical cancer that are not PD-L1 positive, but have TMB high, and we're able to get access to immunotherapy in that setting.
And as a follow-up to that, now when you have, and we are seeing patients that have a complete response after the combination of the carbotaxol, bevacizumab, and pembro, in those patients, are you routinely following in other words, your maintenance therapy with both bevacizumab and pembrolizumab, or just pembrolizumab?
I do both, because the trial was designed like that. So if I started bevacizumab in the beginning, I continue with both drugs later on, unless they have a side effect that makes me stop one of the drugs, but I continue with both as maintenance.
Fantastic. Well, I want to be respectful of your time, Anio, as well, and I know that certainly there were many who joined, and I want to thank all of you who joined. If there are any additional questions that you might have, we'd be happy to address those. Definitely want to be very, very thankful to you and to Rob and to Antonio for such a fantastic job on a special issue. Super proud of having you join us for this occasion. And I also like to announce, although we're a little bit far from it, but the special issue of March of 2024 We have already had the guest editor selection, and it's going to be very, very exciting special issue. This one's going to be a little bit more focused on surgery. So we're looking forward to working with everyone on that. And I want to thank everyone who joined as well. So really very, very grateful. And thank you, Ainhoa, for staying till the end of the Journal Club.
Thank you for the invitation. It was great to be here.
All right. Well, thanks, everybody.
Bye.
Goodbye.